Hello, lightning biologist. I hope all of you are doing well. Today we're going to go over some DNA, what its structure is. Hopefully this is a review if you've been using the other videos and readings and maybe even did the DNA coloring. Let's start just with number one. What does DNA stand for? And so DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, and so you can see the D, the R for ribose, and the A for the fact that it is an acid. What are the two main functions of DNA? Well, DNA, as I'm sure you have heard, is the blueprint of life. And so DNA stores information, and DNA also is able to transfer that information as hereditary information from one individual to its offspring. And so we will just say that it transfers information, and that information is hereditary information that we'll talk about hopefully when we're all back together. A monomer of nucleic acid would be called a nucleotide. And there are two polymers. We have DNA, which we're talking about right now, and we also will talk about RNA, which, by the way, this coronavirus, COVID-19, is actually an RNA virus. And so it doesn't have DNA in it, it actually has RNA in it. How many strands does DNA have? And so if you think about the strand of DNA, hopefully you've colored the strand of DNA this week. DNA has two strands. We say that it is double, or it has two strands. And its shape is known as a helix. And so if I were to draw DNA, I would draw it like a twisted ladder, and then in between would be all of the rungs. And so that's the basic shape of the DNA molecule. It's a really large molecule. Rosalind Franklin actually is the person who we really like to give credit to for discovering or helping Watson and Crick to discover the general shape of DNA. And if you look right here at this image, this is the image that actually allowed Watson and Crick to be able to say, to be able to come up with what the structure of the DNA molecule was. So Rosalind Franklin's X-ray diffraction image was used by Watson and Crick to discover the structure of DNA. DNA in eukaryotes, such as you, would be found in the nucleus. And DNA in prokaryotes, so single-cell organisms, bacteria, is found just free inside the cell because they don't have nuclei. So if we're talking about the monomer of DNA, it's known as a nucleotide. And so this nucleotide, if we think about a nucleotide, there are three parts to the nucleotide. So the first part would be a sugar. The second part would be a phosphate. And then the last is a nitrogen base. DNA is actually pretty simple in well, it's very complex, but yet it's simple in how it's put together. It's a language that's universal. However, there's only 4 letters in DNA because there's 4 different nitrogen bases. And so if I were to ask you to draw a nucleotide of DNA, that would look something like this. You would have your phosphate, and then the phosphate would be attached to this 5 carbon, so I'll draw it with 5 sides, this 5 carbon sugar. And then the 5 carbon sugar would be attached to, and I'm not going to make it quite so beautiful as, I know, the picture right here, because right here would be a nucleotide. So you have your phosphate, you have your 5 carbon sugar, and those are going to repeat. The 5 carbon sugar will be attached to 2 phosphates, and it forms a backbone to protect the important information, which is actually this nitrogen base. Kind of like a book. If I have the hardcover of a book and I keep it shut, it protects what's on the inside, the important information. So if I look at this drawing here, we move up, and I'm going to draw lines linking the following terms to their location on the DNA diagram to the right. Hopefully this is a little bit easier now that I just kind of showed you and drew it for you, but we will do phosphate. We'll have the phosphate. It doesn't look very good, does it? I'll erase that. We'll have the phosphate in red. So right here is the phosphate. I'll go ahead and color that in. We'll make the deoxyribose sugar. We'll make that blue. And as I said before, there's a backbone here of phosphate, sugar, phosphate, sugar. So all of these that I'm coloring in right now, these are all 5 carbon sugars, and they are attached by these phosphate groups that I'll color in red. And on the other side, because it's a double strand, you have the same backbone. You have these phosphates, and in each nucleotide you have phosphate, sugar, and the base. And these phosphates and these sugars attach together to form what we call the backbone of DNA. The last part of the nucleotide, we'll go ahead and let's make that yellow. We'll go ahead and make that yellow. The nitrogen bases are on the inside here, and so this is a nitrogen base. Adenine, it actually is attached to this nitrogen base. And so nitrogen base are all on the inside. And if you notice, there's a pattern there, and it's really an important pattern that are only certain bases can hydrogen bond to each other. The hydrogen bonds then are these bonds right here. They're the bonds that are between the G's and the C's and the A's and the T's. And so when we kind of go back and we think about, so look at this diagram. The T's and the A's are always connected. The C's and the G's are always connected. There's an individual by the name of Chargaff, and Chargaff's rule, along with Rosalind Franklin's image, helped us to understand that the adenine and the thymine are always connected, and the cytosine and the guanine are always connected, and there's no exceptions there. It doesn't fit together any other way. And so the G, which is guanine, always pairs with C for cytosine. And the T always pairs with A, adenine. And that T is thymine. All right, so one way to remember this is if you think about apples. Apples. Pretty bad apple, but we'll go with it. We'll call that our apple. Apples, so A, are always found on, you guessed it, what are apples always found on? Well, they are always found on trees. So A and T always bond together. Then if we think about a car, if I can draw a car for you. Not sure that's a good possibility in my book, but we'll try. So if I'm going to draw this little car here, all right, this car, which is a C, it always is going to go home and drive into the garage. So the garage. So that's one way that I like to remember. Apples grow on trees, A and T, adenine and thymine. And cars, we like to be able to park them in the garage. And so we connect C and G. This helps us because when we get to the next lesson, which is going to be replication, we're going to know that an A will always match up with a T and a C will always match up with a G. The backbone of the double helix is alternating, and it's alternating the sugar and the phosphate. What bond holds these complementary bases together? We know that as a hydrogen bond. It's strong enough to keep them together, but it will not, it can be broken, so that it can be opened up and we can actually read the information. In a DNA molecule, the number of cytosines will always equal the number, well, cytosine, cars go in a garage, cytosine will always equal the number of guanine. And in a DNA molecule, the number of adenines will always equal to the number of thymine. When we're looking at this information, it's the actual information, the nitrogen bases, that tell us the information, or what we would call the genetic code. And so what part of the nucleotide contains the genetic code? It is the nitrogen bases. The sequence of nitrogen bases is similar for all organisms. We read these bases in groups of 3. And so in a word in DNA, it might be A. So adenine, adenine, adenine. On the other side of that DNA strand, then we would know it would have been T, 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 and so on. And we'll talk more about that later on. So this is the basics of understanding structure of DNA, that deoxyribonucleic acid. Hope you enjoyed this. Hope it helped. Stay healthy, stay home, wash your hands. Until next time.